My name is Xin Gao. I'm an associate professor of computer science. I'm also a PI in Computational Bioscience Research Center at KAUST. The title of the paper is a deep learning framework to predict binding preference of RNA constituents on protein structure surface. So protein and RNA interactions are behind all the post-transcriptional regulations, which includes alternative splicing and alternative polyadenylation. And these kind of interactions can regulate and control the expression of different transcripts of genes. Thus, they play very important roles in our cells. The well-known examples include organelle proteins, which is the key player in RNA interference, as well as CRISPR-Cas9 systems. So previously, people have been trying to measure the protein-RNA interactions through two approaches. The first one is experimental approach, for which there are mainly two methods to do the work. The first one is assay-based methods, for which they try to get these high-throughput experiments to measure the specificity of protein-RNA interactions. And such methods can give you a statistical profile of the protein-RNA interaction, but they cannot reveal how proteins and RNAs interact with each other. So you cannot obtain any structure information from such experiments. And another main approach is to do structural biology experiments, for which you are basically solving some snapshots of a protein and an RNA. But from such methods, you can only see those snapshots for which you will not obtain any statistical information of their interactions. On the other hand, the computational community has been trying to solve the same problem by developing some computational methods to tackle it. However, the state-of-the-art computational methods, they mainly do some low-resolution predictions, which means they can only predict for any amino acid of a given protein sequence how likely it is to bind to an RNA. So basically, it is a binary classification problem that the computational community has been trying to solve. And in this paper, we proposed a novel deep learning framework called NuclearNet, which is actually first of its kind that can provide high-resolution predictions for protein-RNA interactions. So what our model can do is, our model is trained on all the known structural biology experiments stored in Protein Data Bank, PDB, from which we tried to learn the specific patterns between protein structure and RNA sequences. And for that, our model can accurately predict the binding specificity and binding preference of all the high-resolution RNA constituents on any location of any protein structure. So given a query protein structure, NuclearNet can actually predict for any location on the surface the binding preference of ribose, phosphate, and 4 different bases. And how does this help researchers? Well, researchers can, number one, visualize our results, for which they can derive the prediction on whether this particular query protein is an RNA-binding protein, or which is known as RBP. And if so, where is the most likely binding pocket on the surface of this protein? And furthermore, what is the most likely binding RNA sequence for this pocket? And this can help them to, number one, for a known RBP, to design the best RNA sequence that can interact with this RBP. And number two, for unknown RBPs, can help them predict and identify the potentially new RBPs. So our method has an open web server that the entire community can use, and I hope you like it. Thank you.